So this video is on the topic of experimental design, which is something you should be pretty familiar with after taking science for several years. In order to make sure that the results of your experiment are valid, in other words, that your data that you get from the experiment actually answers your question and gives you a reliable conclusion, you need to make sure that the experiment contains the following things. The first is that it contains one and only one independent variable. The second is that it contains a dependent variable. You can actually have more than one of these, but for the most part, you're really only going to have one dependent variable. The third is that it has controlled or constant variables, and there's usually a lot of these in an experiment. And the last is a control group, which is something that you compare the rest of your data to. A control group is not necessary for a valid experiment, but usually the best experiments have some sort of control group. So in an experiment, the independent variable is the variable that you change in order to see how it affects everything else in an experiment. Because you're able to change it, it's often called the manipulated variable in an experiment. So let's say you're doing an experiment to figure out how the amount of water affects the height that a plant will grow to. In this experiment, because you're changing the amount of water is your independent variable. In any valid experiment, you're only really allowed to have one independent variable, which means that you can't change other things like the amount of light, the amount of soil, any of that all has to be held the same. The other thing is you want to have multiple levels of your independent variable. So if we're changing the amount of water on plant height, you might give one plant 0 milliliters of water, you might give one plant 10 milliliters of water every day, and you might give another plant 20 milliliters of water every day, and see how those affect plant height. In the same experiment, you're also going to have a dependent variable, which is what you're measuring. So in this case, if we're measuring the amount of water on plant height, the thing we're actually going to measure is the height of the plant, and you'd probably do so with a ruler. The other thing you're going to need in this experiment is something called controlled variables. Controlled variables are also called constant variables. These are the things that you keep the same throughout the experiment. If you have multiple things that you're changing, you're not going to know the impact of your independent variable. Therefore, you have to have constant variables. In this case, constant variables would be things like the amount of light your plants are getting. So there's the sun for amount of light. They might be that you have the same type of plant, so these are all the same. They might be that they're in the same size pot, which these are all the same size pot, or that they have the same soil or fertilizer. Even things that you don't think about are constant variables. For example, a lot of times we do experiments in the same room so that the room temperature is constant, or we use the same source of tap water so that the quality of the tap water is constant. We really try to keep everything the same in an experiment so that the only thing that's changing is the independent variable. Finally, the last thing in a valid experiment is the control group, and it's really important that you don't confuse the control group with controlled or constant variables. The control group is the group where no treatment is done to it, so they get kind of a 0 level of the independent variable. In this case, when we're measuring the amount of water on plant height, the group that gets 0 water, which is this plant right here, this is our control group. And really, we use the control group as a comparison to see how the amount of water changes. Once again, you don't have to have a control group to make it a valid experiment, but it does help your results as something to compare it to. Finally, I want to push for the idea of trials. In an experiment, if you only do one trial or you only do an experiment one time, you're not really sure that the results are valid. Remember that an experiment has to be repeatable and testable. So the more trials you have, the more data you have. And the more data you have, the more likely that your results are correct. It's easy to make a mistake in one trial, but it's hard to make the same mistake over and over and over again between different trials. After the experiment, 2 things happen. Number 1, you analyze your data, and number 2, you're able to draw a conclusion. We'll talk in the next little module about how to analyze data through graphs and tables and how to do it properly. And we've also talked a little bit about how to draw a conclusion before and what goes into a valid conclusion. But remember, your conclusion isn't going to be valid unless your experimental results are valid, and that's only going to be valid if your experimental design is valid and doesn't have errors in the process. So what are the big takeaways from experimental design? The biggest is that you can only have one independent variable to have valid results. The second is that in order to only have one independent variable, you have to control all of your other variables, and this sometimes is very challenging to do in a lab setting. Finally, the more trials you'll have, the better the data you'll collect. So that's it for experimental design. You'll have multiple opportunities throughout this course to actually design experimental protocols and talk about strengths and weaknesses of experimental design, but it's really important that you remember the basics so that you don't make mistakes in procedure.